Today we're going to explore one of the most fundamental ideas in all of science, the unit of life. So, let's start with a question. What could you, a massive, towering redwood tree, and the tiniest little bacterium possibly have in common? I mean, on the surface, absolutely nothing. But if you zoom all the way in, way down to the microscopic level, you all share the exact same building block. And that building block, of course, is the cell. Now, the official definition calls it the fundamental, structural, and functional unit of all living organisms. And that's a pretty big deal. It means every single living thing you can think of is built from cells and runs on cells. It's not just a part of life, it is the unit of life.
But, we didn't always know this. It actually took centuries of brilliant detective work to figure it out. The story kind of starts way back in 1665, when a guy named Robert Hooke looked at cork under a microscope and saw these little boxy shapes he called cells. A few years later, Anton von Leeuwenhoek was the first person to see a living, moving cell. But the real game changer came in the 1800s. First, Schleiden and Schwann realized, hey, all plants and animals are made of cells. And then Rudolf Virchow laid down the final, crucial piece of the puzzle. All cells have to come from other cells.
So, all of that amazing history can be boiled down to two really simple, really powerful rules. We call it the cell theory. Number one, all living things are made of cells. And number two, all cells come from other cells that already exist. That's it. Simple, elegant, and the absolute foundation of modern biology. Okay, so all life is made of cells. But here's the twist. Not all cells are built the same way. In fact, nature came up with two basic designs, this great divide that splits all life on Earth into two huge categories. And this, right here, this is the single biggest difference between them. On one side, you've got prokaryotic cells. Their genetic material just kind of floats around freely inside. But on the other side, you've got eukaryotic cells, the kind you have, which keep their precious DNA locked away safely inside a membrane-bound nucleus. And that one little architectural change, it changes everything. So let's start with the original model, the ancient blueprint for all life that got the whole party started, the prokaryotic cell. These were the true pioneers of life on this planet. So when you think prokaryote, think bacteria. These guys are like the ultimate minimalists. They're stripped down, super efficient little life forms. They're usually way smaller, they can multiply like crazy, and they don't have any of the complex internal compartments or organelles that other cells do. Their DNA is just one simple circular loop called a nucleoid. Minimalist, but incredibly effective. Okay, now let's check out the upgraded model. The eukaryotic cell is all about organization and having specialized compartments. I like to think of it like a busy city with all these different buildings and factories, and each one has a very specific job to do. And the two most famous examples of this cellular city are plant cells and animal cells. Now, you can see right away they share a lot of the same basic machinery, but you can probably also spot some pretty big differences. So let's take a little tour, shall we? Every city needs a border, right? A city limit. And for the cell, that's the plasma membrane. But what's so cool about it is that it's not a rigid wall. It's what scientists call a fluid mosaic. It's this flexible, flowing barrier that acts like a very picky gatekeeper, carefully controlling everything that goes in and out. So how does stuff actually get across this border? Well, there are basically two ways. The easy way is called passive transport. That's when molecules just naturally flow from where there's a lot of them to where there's less of them. No energy needed. But sometimes the cell needs to move something against that flow, from low to high. That takes work. That's active transport, and it requires the cell to spend some energy in the form of a molecule called ATP. Right at the very heart of our eukaryotic city is the command center, City Hall itself, the nucleus. This is where the cell keeps its most valuable possession, all of its DNA, which is neatly packaged into these structures called chromosomes. All the blueprints for building and running the entire cell are kept safe right here. Now, of course, a city needs power to run, and that very important job goes to the mitochondria. You've probably heard these called the powerhouses of the cell, and it's no joke, that's exactly what they are. They take in fuel like sugars and perform cellular respiration to crank out tons of that ATP energy we were just talking about. Next up, let's visit the city's manufacturing district, the Endoplasmic Reticulum, or just ER for short. You can see it has two parts. The rough ER is studded with these little dots called ribosomes, and its job is to build proteins. The smooth ER, which doesn't have ribosomes, specializes in making things like lipids. So once all those proteins and lipids are made in the ER, they're not quite ready to go. They need to be processed, sorted, and shipped to the right destination. And that is the job of the Golgi apparatus. It's basically the cell's post office. It takes in the packages from the ER, makes some final tweaks, slaps a shipping label on them, and sends them on their way. All right, here's a feature that's exclusive to the plant cell city. If mitochondria are the power plants that burn fuel, then chloroplasts, well, they're the solar panels. These amazing organelles are where photosynthesis happens. They capture the energy from sunlight and turn it into food, which is the energy source for almost all life on this planet.
Whew, okay, that was a whirlwind tour. We've covered a ton of ground. So let's just pull everything back together and put the key differences side by side. You know, a little tail of the tape.
So the great divide, right? Prokaryotes versus eukaryotes. It really comes down to this. Prokaryotes are the minimalists. They're smaller, they have no nucleus, their DNA is a simple circle, and they don't have all those specialized organelles. Eukaryotes, on the other hand, are all about complexity. They're bigger, their DNA is protected inside a nucleus, and they've got that whole suite of organelles working together like a well-oiled machine.
And even within the eukaryotes, what's the deal with plants versus animals? Well, their differences are all about their lifestyle. Plant cells need to be rigid, so they have a strong cell wall. They need to make their own food, so they have chloroplasts, and they need to store water, so they have a big central vacuole. Animal cells are more mobile, so they don't have those things, but they do have structures like centrioles that help out during cell division.
And this really gets at the power of this whole idea. The scale here is just mind-blowing. You can have a tiny Mycoplasma bacterium that's only 0.3 micrometers long, or you can have the single massive cell that is an ostrich egg. And yet, the fundamental blueprint, the cell, is the same universal foundation.
So I'll leave you with this to think about. A single bacterium, one prokaryotic cell, is a complete self-sufficient living thing. A human, on the other hand, is a cooperative of trillions of specialized eukaryotic cells all working together. So what does that profound difference in scale and organization really tell us about what it even means to be alive?